Alright, 7th graders, I just wanted to give you a companion video to go to with your WebQuest. So if you're at school and you're having issues getting onto some of the Adobe Shockwave sites that you need to access and finish the WebQuest, if you have this video up and the WebQuest up at the same time, you should be able to do it. So the first one is going to the incomplete dominance game or tutorial, and if I'm going to go through this, I'm going to read it to you so I can know my pacing. So it says, in the 1800s, Gregor Mendel conducted experiments with pea plants like the ones shown above. He discovered some of the basic rules of inheritance that we still use to understand genetics today. Mendel observed that some traits appeared to cancel out others and called these traits dominant. He called traits that seemed to be canceled out recessive. Try crossing some pea plants to figure out which flower color trait is dominant. So let's try a white and a purple. When I hit cross, all of them come out purple, so keep that in mind. Let's try a purple and a purple. When I do a purple and a purple, again, I get all purples. Now let's see what happens when I get white and white. So when I cross white and white, they all are white. So that shows us that one color is probably dominant than the other. And let's see if your hypothesis, which color you think is dominant, is correct. Each pea plant has 2 copies of the gene for flower color, 1 on each chromosome. And you get 1 chromosome from your mom and 1 from your dad. But different forms of the genes, called alleles, might occur on each chromosome. In this case, each chromosome might have an allele for purple flowers, and see, that's a capital P. And when you talk about genetics, the capital and lowercase absolutely matters. Or white flowers, lowercase p. Use the pull-down menus up here, so we're going to change that. So let's see. Right now we have capital P, and that gives us purple flowers. What happens if we do little p and big P? We still have purple flowers. What happens if we do little p and little p? Oh, now they're white. So the appearance of capital P, so capital P gives us purple. Capital P and little p gives us purple as well. But when we do little p and little p, we get white. So you should know at this point, hopefully, which flower color is dominant. And on that one, purple was dominant, white was recessive. Now we're going to look at these snapdragon plants. We're going to try crossing them like we did with the pea plants to figure out which allele for flower color is dominant. So if we do pink and we do red, wow, we get both pink and red. So they're not all one color. We have different colors. Let's see what happens when we do pink and white. When we do pink and white, we get some white and some pink. Interesting. Let's see what happens when we do 2 pinks. Oh my goodness. Some are pink, some are red, and some are white. What happens when we do both red? And sometimes this app shuts down and we have to just go back and start. That's okay. So when we do 2 reds, we get all red. So knowing that, I'm going to hypothesize when I do 2 whites, I'm going to get all white. And that's true. So do we have just one dominant color? Some of the snapdragons had different colored flowers than you would expect, didn't they? Unlike the peas that had 2 different flower color possibilities, there were 3 different flower colors, red, white, and pink. Did you figure out why? So this is where the vocabulary comes in. And the 3 main themes we're thinking of in this is dominance, incomplete dominance, and codominance. So we're going to see what we think the snapdragons were. All right, lastly, on this one, here's some lentil plants. So let's look at the seeds. So the seeds with the larger areas of color are called spotted. Those are these guys. Or if I move my mouse over, I've got some small points of colors which are called dotted. Okay? And then look, I have some that are both spotted and dominant. Let's see what happens when I cross a big one and a big one. They're all big. Okay. What happens when I cross a spotted and a spotted? They're all just spotted. What happens when I cross a dotted and a spotted? I get all, mixed, dotted and spotted. What happens if I do a dotted and spotted with a dotted and spotted? Ooh, interesting. Some are just spotted, some are dotted, and some are mixed. What happens if I do a spotted and a mixed? I get some that are mixed and some that are spotted, but none that are just dotted. Let's see what happens when I do, nope, it's going to make me refresh. Let's see what happens when I do just, okay, when I just do the dotted, I get all just dotted. But what happens when I do the mixed and the dotted? I get some that are mixed and some that are dotted, but none that are the big spotted. Okay, do we have one that's dominant? If so, which one do you think it is? All right, so what happened when you crossed a plant with spotted seeds, that's this guy, with one that had dotted seeds, that's this guy? If the trait for the pattern of the seeds on the lentil plants followed Mendel's rules of dominance, the seeds would always either have just one or the other, like the plant shown above. Instead, a distinctly different appearance, spotted and dotted, or what I was calling mixed, can occur. That's because lentil seed pattern is an example of codominance. This means that when an individual receives two different alleles, one for spotted and another for dotted, they are both expressed fully, producing a seed with spots and dots. But do you think this explanation works for snapdragon flower color too? Let's see. Snapdragon flower color does not follow Mendel's rules of dominance either. When a snapdragon receives two different alleles for flower color, one for red and another for white, like in this example up here, they both influence its appearance. Rather than showing like half red and half white like we saw in the codominance, an intermediate or blended appearance that is different from both parents, pink flowers, is created. This is called incomplete dominance. Okay, blood type in humans is determined by what type of sugar polymer is on the blood cell surface. If I click up on the microscope, look, this blood type shows A. That means that all the alleles for that blood type have A. That's one parent has A. If I look at the next parent, they have the alleles capital B. What do you think their parents are going to have, or their offspring is going to have? They have mixed AB. Do you think that's dominance, incomplete dominance, or codominance? So this is kind of like them having both, so this would be our codominance. Blood type in humans is an example of codominance because the AB blood type has the characteristics of both A and B. Okay, rabbits can have fur of different colors. When this white rabbit mated with a brown rabbit, all of the offspring have brown fur. What kind of dominance do you think fur color in rabbits has? So hopefully when you thought about it for a second, you picked dominance because that's when we have either or. Fur color in rabbits is an example of dominance. The allele for brown fur is dominant because it appears to cancel out the allele for white fur. Horses can have different colored coats too. When this cream horse mated with this brown horse, their offspring had a tan coat. This is called a palomino. What type of dominance do you think coat color in horses has? And this should remind you immediately of the incomplete dominance that our snapdragons had when the red and the white made pink. So coat color in horses is an example of incomplete dominance. The offspring had an intermediate blended appearance that was different from both parents. And that's it for this one. We're going to move on to the second one. Okay, we're going to do the next interactive game on Punnett squares. So let's go ahead and start. It says your friend has a pet chicken and wants to breed it so she can raise the chicks. She has found a rooster for her hen to breed with, but what she wants to know what their chicks will look like before she breeds them. How can we predict what the offspring of her hen and this rooster will look like? We need to know what outcomes are possible. In order to figure out all of the possible genotypes that can result from crossing two organisms, scientists sometimes use a tool called a Punnett square. This table is called a Punnett square. A Punnett square lets you display all of the possible combination of alleles that will result from a cross. At the top and left, you can see the two chickens we are going to cross. Your friend especially wants brown chickens. Capital B and little b are the two alleles for this gene. You can see the alleles of each chicken listed near their picture. Capital B is dominant, producing brown feathers, and little b is recessive, resulting in yellow feathers. How do you think the Punnett square might help us find all the possible genotypes of the chickens? As you can see, the two alleles for the colored gene from each parent have been separated and given a row or a column. So remember, rows go from left to right, columns go from up and down. Following these rows and columns, you can see how the Punnett square is used to find all the possible combination of alleles from the two parents. Each box in the square contains the product of a cross between a row and a column. So if we cross this row right here from the dad. He's a rooster, so he's the dad. This is a mama. If we cross this row, capital B, see it's right here, and this column, capital B, we are going to have the genotype capital B, which is going to give us brown chickens. Excuse me, if we look at this one, we bring this row all the way across, and there's my capital B. We bring my little b down for a genotype of big B, little b, which again is going to give us brown chickens. Again, if we look at the bottom of the Punnett square, if we bring the little b in, and we bring the big B down, we have little b, big B, which is still going to give us brown chickens because capital B, or brown, is dominant over yellow. If we look here, we're going rows then columns. Little b, together, that gives us the recessive genotype, or yellow feathers. So looking at the boxes, it's easy to see that some allele combinations show up more frequently than others. This means that on average, these genotypes are more likely to be produced when these two chickens mate than other combinations of alleles. In this case, the cross has produced one box with capital B, two that hold capital B, or little b and big B, and one with little b. In other words, the ratio of genotypes is 1 to 2 to 1, or 1 homozygous for the dominant allele, capital B, 2 heterozygous with the genotype capital B or little b, and 1 homozygous for the recessive allele, little b. Remembering that big B is the dominant allele for brown feather color in chickens and little b is recessive, how often would we expect to see brown chicks if your friend breeds these two chicks? So look, we look big. If there's a big B, it's always going to be brown. So there's a big B there, there's a big B here, and there's a big B here, no big B here. So out of the 4 squares, 3 of them are going to be brown. So a chicken will show the trait and look brown even if it only has 1 copy of big B, since it is the dominant allele. In this cross, 3 of the 4 boxes contain the capital B allele. 1 out of 4 times the offspring will have the genotype capital B, meaning it's homozygous, but 2 out of the 4 times, or half the time, they will have the genotype capital B or little b, heterozygous. Offsprings with both these genotypes will display the dominant trait. So the answer is 1/4 plus 2/4, or 3/4 of the time. Now fill in your own Punnett square. It is set up for a cross between these two lemmings. Their respective alleles for a fur color gene are labeled on the sides of the square. The 2 alleles are capital A and little a, and you can see from the pictures that both of these lemmings are capital A, little a, or heterozygous. Since the alleles that should go into each base, select the alleles. So if we look at this one, remember we're bringing a row over capital A, would give us big A. Bring this one over big A and little a, it's going to give us this. Bring this row over little a, bring this down big A, give us this, but really these two are interchangeable, it doesn't matter. We bring this row over, we get little a, we bring this row over, we get little a, and that should be right. So we are correct. Suppose that the gene in the 2 lemmings you just crossed is the gene that causes albino offspring. Albinism is a condition in which the normal pigment of the skin, fur, and even eyes is absent. It is also a recessive trait. Looking at the results of your Punnett square, how many crosses resulted in lemmings with genotypes that have the allele for albinism but won't be albino? These individuals are sometimes called carrier. So we need to look at the guys that are not albinos, and the ones that are albinos are just this one because they have to have that both recessive alleles. These guys don't have any little a's, so they're not going to carry it either. But look at these heterozygous guys. Even though they're not albino, because they don't have both recessive alleles, they each carry a recessive allele. So we're going to have a chance of 2 of them being carriers. Yes, so since albinism is a recessive trait, only individuals with the genotypes little a will be albinos. However, all of the heterozygous offspring, genotypes capital A, or little a, capital A, will have the recessive allele without being albinos. They are the carriers. What are the chances that a carrier will be produced? Because we had 2 out of the 4 boxes in the Punnett square that have these genotypes, there's a 50% chance that a carrier will be produced each time these organisms have an offspring. Suppose that you are breeding these lemmings and want to produce albino offspring. Looking at the Punnett square again, you know that there's a 50% chance that a brown colored carrier will be produced and a 25% chance that an albino lemming will be produced with these parents. But that's not good enough for the high-stake worlds of lemming breeding. How can you make the chance that an albino offspring will be produced better than 25%? One way is to carefully choose the lemmings you breed. Choose 2 lemmings from the litter that resulted from the last cross by clicking on them. So in this one we did, I have to probably refresh this. Go through this really quick. Alright, and we said this is 2. Alright, so we had to wait. Okay, it wouldn't let me highlight them, so we did it anyway. So let's try to give us a better than a 25% chance. Yes, it's going to be those. Alright. One way is to carefully choose. Okay. Hello. So the 2 lemmings that we selected are ready. Fill in the Punnett square for them. Alright, so we've got big A here, little a. So we're going to have this one. We bring this one over. We've got big A and little a. So it's going to be this one. This one we're going to have little a, and this one we're going to have little a and little a. So we did it correctly. We're going to see that we can get our albinos more than 25% of the time. So we have a 50% chance of getting brown lemmings and 50% chance of getting albino lemmings. Our last challenge is to fill a very special order, a long-tailed albino lemming. These are very rare, and it's going to require all of our skills to produce one. The computer will keep track of how many tries it takes you, but don't worry. Okay, so we're going to do it with our Punnett squares. Here's our stock of lemmings. Note that some of them have long tails and some of them have short tails. The allele for long tails is capital L is dominant, and the allele for short tails, little l, is recessive. But where are the albino lemmings? Remember that lemmings that only have 1 copy of the recessive allele for albinism won't look albino. So we have to pick 2. So this guy has, let's just try it. We'll try 1, or we want long-tailed ones, right? So we'll pick 1 and 4. Okay, so they're going to breed. We got to let them have their privacy there. The litter of lemmings did not produce an albino with a long tail, okay? So none of them did. So they all had long tails. Ooh, look, some of them had short tails, which means they were carrying the recessive allele for short tails. Let's see next time. Here's my stock of lemmings again. Let's see. Let's try this long tail guy and this long tail guy. Ooh, goodness. All right, let's see how it looks. So he has the allele, he's heterozygous for long tail, meaning he carries an allele for long tail and one for short tail, but the long tail's going to be dominant. So this guy, lemming number 3, he's got both dominant alleles. So that means every single one of these offspring, even though they have some of them have the short tail allele from this parent, they're all going to have long tails. So that's good. But here's the problem. This guy has both dominant alleles for color, so they're all going to be brown, and this guy has the dominant as well, even though he carries a recessive allele. So none of these are going to be albino again, so we don't want to breed those. But we might want to hang out with number 3 again. Let's see. So let's try 3 and 1. Let's see what 3 and 1 happen. Okay, now we have long tail, we have long, so all of our lemmings are going to have long tails, which is what we want. Now we're worried about the albino. Because both of them carry the albinism trait, we have some that are going to be albino. We actually have 4 that are going to be albino. So let's go ahead and try to breed those and see what we get. We managed to complete the challenge, okay? So we got 1 baby albino lemming, which is awesome. If this works at your computer at home, please go play the game and try it. So Punnett squares are very useful ways to determine all the possible outcomes of a cross. They can also help us determine the chances of a particular combination of alleles occurring by showing us the ratio of the genotypes. But Punnett squares can also be extended to help us understand the genotypes of whole population. And you probably won't do that until you get to high school. Hold tight, let's do just kidding, there is no last one. It used the same simulation that we just used. So go back and use it to do your questions on albinism that you have at the bottom with your lemmings. And that should be it. Just those two videos or those two simulations are the two that you need to get through your WebQuest. Have a good day.